Apakro. I think now we are going for another session, which would be brief only, nothing to worry. So, the second topic would be recent advancements in tuberculosis. It is encompasses three components. One is the drugs, the second one is diagnostics, the third one is miscellaneous part of it. So, coming to the basic epidemiological graph, what we see. So, we want to have an end TB strategy by 2035, as envisaged in the WHO document with reference to Indian perspective. We can see the mortality as well as the incidence. Incidence is gradually coming down from anywhere between 22, 23 to around 3 to 5 by 2035. That's what the projection. And the mortality is also coming down more or less in parallel with the interventions called better and shorter treatment, affordable care, novel diagnostics, and lastly the prevention, which is the most important factor with reference to Indian perspective. So coming to the outline, with reference to treatment, just now we were discussing the drug-resistant TB and drug-susceptible TB. Now we are moving from drug-susceptible to drug-resistant TB because we claim ourselves that we have conquered the drug-sensitive TB with the drugs that are available in the market. So coming to the diagnostics, various rapid diagnostic tests for detection of MDR TB and latent TB also. And last comes the prevention, that is vaccine under development. So coming to the advancements with reference to drugs, we have various drugs with reference to the MDR TB. So in this chain we have delaminate, and we have the investigation drugs like SQ109 and bedaquiline, and we have the BA824 and sidjolid. These are the various drugs with various actions at the molecular level. So coming to some of the drugs, just I'll quickly run through. These are, some of these drugs are US FDA approved. Bedaquiline in the dose of 400 milligrams daily for first 2 weeks, followed by 200 milligrams 3 times per week for 22 weeks. The main approach with all these new drugs is to cut down the duration of therapy, because duration is very longer, the compliance is also at risk. Next is, it shortens the time to culture conversion and no cross resistance with the standard drugs that we use, that is ethambutol, pyrazinamide, and streptomycin and other aminoglycosides, and the common quinolone, that is moxifloxacin. So the precautions that we have to take are, one is it prolongs the QT interval, and frequent monitoring with ECG is very much required, and we have to monitor the liver function tests also, just like any other drug. No adverse drug interactions with other anti-TB drugs that commonly used. And compared to the placebo, it has 2.5 higher mortality rate because of this QT prolonged interval. Should be used only when an effective regimen cannot otherwise be provided to the patient. So this is a precondition that they have put forth in the WHO. So coming to various novel combinations to treat MDR TB, we have various drugs by the WHO, bedaquiline, moxifloxacin, and pyrazinamide, PA824. Just now we were telling some of the investigation drugs. They are in the phase 3 and phase 2 trials, most of them. And we have this, for MDR, we have this phase 3 trial, 24 months conventional regimen, moxifloxacin, pyrazinamide, kanamycin, ethionamide, and teridone, versus 6 months of investigation regimen. So they want to bring it down to 6 months from 24 months, which is a quite lengthy regimen to treat with for MDR. So the other drug is delaminate. It is also approved by European Union, but it is not yet approved in the US FDA or by the Drug Control General in India. It is undergoing phase 3 trial in the United States. So the other one is Tuberculosis. It is another linezolid analog. Of late, linezolid is being used in the MDR in the 600 milligrams BD dose. It has a better antimicrobacterial activity and improved safety profile when compared to the linezolid, which is an anti-staphylococcal drug. It has potential to shorten the treatment duration. That's very important. And currently it is in the phase 2 trial. So this is another, which is an analog of ethambutol, SQ109. It is active as drug susceptible as well as MDR strains. Early bactericidal action when compared to the ethambutol, which is a static action. And it is synergistic or additive with reference to INH, rifampicin and second line antibiotics. It has proved that it has a low bacterial mutation rate. It is also undergoing phase 3 clinical trials with reference to MDR-TB. So the other one is proteaminate. So it's a potent bactericidal and with the sterilizing action. That's very, very important to wipe out the dormant bacilli. And it's safe, well tolerated with a small dose of 100 to 200 milligrams. It has got some synergistic effect with reference to moxifloxacin and pyrazinamide, and it has a potential to shorten the treatment. It has no significant interaction with reference to CYP450 stimulation. Useful option for HIV/TB co-infection. Currently, it is also undergoing. They use better bactericidal activity than standard control regimen, shortening to 4 months. It's not that effective, but no significant difference in the adverse effects. So 6 months remains the gold standard for tuberculosis management. So study carried out in 416 patients, the newly diagnosed from positive, and 4 months thrice weekly regimen, that is intermittent chemotherapy, with moxifloxacin/gatifloxacin, another drug, with HRZ was inferior to standard 6 months treatment. So we have one more called STAND trial, that is shortening of treatments by advancing novel drugs. So they wanted to try this PA824 with moxifloxacin pyrazinamide. First trial to test the common regimen as well as the drug resistant bacillus. So what are the problems with rifampicin? The major limitations with rifampicin are low and variable bioavailability, subtherapeutic rifampicin levels, serum rifampicin, poor GI and hepatic tolerance, and multiple drug-drug interactions, and cytochrome P450 induction, and overexpression of FECP, slow response non-adherence treatment, leading to slow response. So this Cadila Pharmaceuticals has come out with a novel intervention or innovation, what they call rifampicin overcoming these limitations. So they have added this piperine. Piperine is an extract from the black pepper. It's a bioenhancer. Piperine. Inhibit cytochrome P450 enzyme, prevent overexpression of TGP, and ensure high and sustained serum rifampicin levels, improves sputum conversion rate, reduce GI effects. So Rizorin is a combination of, it's a FDC, that is a fixed dose combination of rifampicin, INH and piperine. So thereby they want to ensure by adding this, these are the things they are achieving, or these are the effects they are achieving. High and sustained serum rifampicin levels, improves sputum conversion rate, and reduce GI side effects. It's an outcome of joint research by CSIR Center from Jammu, that is IAM, that is Indian Institute of Integrative Medicine. It was approved by Drug Control General of India way back in 2009 for treatment of drug sensitivity. just like just now Dr. Bhaskar was telling, some of the molecules they click, some of the molecules they just sail through. Something like happened here also. Conventional rifampicin versus rizorin. So, on comparison, serum rifampicin levels are only use rifampicin, low and variable. Whereas if we add this rizorin by an answer, the high and sustained. GI tolerance with pure rifampicin is poor because we have a lot of nausea, vomiting, and all that. But that is improved. GI tolerance is very much improved. Risk of hepatitis is very high when compared to the rizorin being added. Reduced risk is there, and adherence is low because of the various side effects and all that, because of poor GI tolerance. Whereas adherence is high because of the improved tolerance. So rizorin clinical efficacy is at par with standard regimen. So what are the benefits of rizorin? Rizorin regimen associated with better tolerability and improved patient compliance. That is very important. It refers to a long drawn anti-TB regimen. Benefits of improved adherence to treatment: lower treatment failure rate, reduced relapse rate, minimized risk of developing drug resistance. These are the outcomes once we have the improved patient compliance. So that's what we want to have it at the end of the day of anti-TB therapy. So with the above facts, rizorin has a place in therapy refers to tuberculosis, that is drug-sensitive tuberculosis. It is a preferred choice in the treatment of drug-sensitive pulmonary, extrapulmonary tuberculosis, particularly in conditions known to decrease rifampicin absorption. Patients who are not likely to tolerate the 450 mg dose, that is 10 mg per kg body average Indian dose. Ideal switchover therapy for patients who develop the intolerable GI side effects or heterotoxic resistance. We have the LPA, no LPA, and we have strict specification that detects specific antigen, that is MPV64. We have the Capilla, rapid TB diagnostics. These are all very expensive, not available everywhere. They are only available in accredited labs across the country. We have the national reference lab and we have the intermediate reference lab in our country. So we have the automatic detection or MDR-TB screening with molecular line probe assay. So we have an IRL, intermediate reference lab in our department that is having this facility of line probe assay. What they do. So we have the gene export also, some of the centers at NRL, and we have the MTB-DR plus, that is drug resistance plus, and MTB-DR SL. So coming to the IGRA, that is interferon gamma release assay. This is also another indirect test. It measures the interferon gamma secreted by T cells that are infected with the organism. And two commercial tests are there: quantiferon TB Gold and T-spot test. Quantiferon TB Gold is one of the most commonly sought after tests by most of the non-test physicians, especially for the extrapulmonary tuberculosis, by ortho, ENT, other people. Blood test measures the cell-mediated immune response, that is interferon gamma using ELISA, and is more accurate and reliable alternative to tuberculin skin test. Can diagnose the TB infection in the latent phase, does not differentiate between active TB and latent TB, unaffected by BCG vaccine. This is one advantage with this quantiferon Gold. So serological tests and their relevance is reference to our country because we have a huge load. We have a huge load of tuberculosis in our country. How far these are acceptable in our context is reference to diagnosis. Detects humoral immune response to MTB. Mainly ELISA 1.5 million tests are performed to diagnose active TB. Approximate cost would be 825 million per year. In July 2011, they issued a policy stating that they are inconsistent, imprecise estimates of the disease. That's why we cannot take them as gold standard for tuberculosis detection. And it's a crime to prescribe as a routine test in each and every case. Go for this serology test. So WHO has categorically mentioned you cannot prescribe it as a routine test. So third component is prevention. The new vaccines are actually needed because it's an age-old BCG. So the current status is, currently BCG is the only available vaccine for tuberculosis. It has a poor predictive efficacy ranges from 0 to 80. As per the Changalpur trial, it is 0 to 80%. Two sets of TB vaccines are under development: BCG replacement vaccine, booster to BCG. This is the latest one, MVA85A, and areas 402 are in advanced stage of development with reference to vaccines. So, coming up, despite all advances, TB remains still a major challenge as far as communicable diseases are concerned, though we have the NCD on the rise with reference to the mortality as well as morbidity. Still, in the communicable disease segment, TB still is the number one. And rapid diagnosis is possible with gene expert, MTB DR plus and MTB DR SL for XDR. This is for XDR. That is extremely drug resistant TB. Newer drugs have potential to improve the outcomes in MDR TB or MDR for treatment of drug sensitive TB